How to see DNA. The traits that you inherit from your parents are determined by DNA. This is true for all living things. The cells of every plant and animal contain DNA molecules. DNA carries the genetic information that determines what the plants and animals look like. Among other traits, you may wonder, though, what does DNA look like? Try this activity to find out. How to see DNA. Purpose. In this activity, you will free DNA from the cells of green split peas. Then, you will be able to see what DNA looks like. You will need half a cup green split peas, 1/8 teaspoon of salt, 1 cup cold water, a blender, a strainer, measuring cups and spoons, liquid detergent, small glass tubes or containers, meat tenderizer, rubbing alcohol, and small wooden stir sticks. Step 1. Blender insanity. Put the split peas, salt, and cold water in a blender. Put the blender lid in place. Blend on high for 15 seconds. The blender separates the pea cells from each other, so you now have really thin pea cell soup. Step 2. Soapy peas. Pour the pea mixture through a strainer into a measuring cup. Add 2 tablespoons of liquid detergent to the strained peas. Swirl the mix. Let the mixture sit for 5 to 10 minutes. Step 3. Enzyme power. Pour the mixture into the glass containers. Fill each container 1/3 full. Then add a pinch of meat tenderizer, enzymes, to each container and stir gently. Be careful. If you stir too hard, you'll break up the DNA, making it harder to see. Step 4. Alcohol separation. Tilt your glass container. Slowly pour rubbing alcohol into the container and down the side. It should form a layer on top of the pea mixture. Continue pouring until you have about the same amount of alcohol as pea mixture in the container. DNA will rise from the layer of pea mixture into the alcohol layer. Use a wooden stick to draw the DNA from the alcohol. After you finish, you will have completed a DNA extraction. Why does this happen? Alcohol is less dense than water, so it floats on top because 2 separate layers are formed. All of the fats, lipids, and protein that you broke up in steps 2 and 3 along with the DNA have to decide, which layer should I go to? Most particles and molecules will stay below the alcohol or dissolve in it. The DNA will float in the alcohol and will not be dissolved. This makes it easy to extract.